Hello, everyone, and welcome to Lunch Break Science. I'm Ariel Johnson from the Leakey Foundation, a nonprofit dedicated to supporting human evolution research and sharing discoveries in programs like this. We want to thank Anne and Gordon Getty and Camilla and George Smith, whose generous support has made this episode of Lunch Break Science possible. I have an exciting announcement to make, but you're going to have to stay tuned till the end of the episode for it to be revealed. Today we have with us 2015 Leakey Foundation Baldwin Fellowship Scholar, Dr. Ainash Childebayeva. Ainash, thank you so much for joining us today.
Thank you for having me. Hi.
Ainash originally hails from Kazakhstan and is joining us from Jena in Germany, where she is currently a postdoctoral student in the Max Planck Institute for the Science of Human History. Is that Science of Human History? Is that the correct institute?
Yes, it's Science of Human History.
Ainash has traveled all over the world studying both current populations and past populations to look at epigenetics, which is what you'll be talking with us about today. Before we hear from Ainash, though, for those of you joining us live on YouTube, Facebook, and Twitter, you can post questions in the chat for Ainash anytime during the episode. The sooner you get those questions in, the more likely Ainash is going to be able to answer them at the end. As I said, please put them in the minute they pop into your head. You can post them anytime during the lecture. My first question for you, Ainash, is, what exactly is epigenetics?
So epigenetics literally means like epi's above, so it's above genetics. So here we talk about any modifications to the DNA that don't change the sequence of the nucleotides, but can affect how genes are expressed. They can either increase or decrease gene expression. And these and why I think epigenetic modifications are interesting is because they can change over time, and they can be they can be associated with various exposures to things like chemicals, as well as someone's diet can affect it, and how much they exercise. So a lot of these factors a lot of the environments that we're exposed to can affect our epigenome.
Now, you study the DNA of both ancient populations and current populations. What are the differences in collecting samples and analyzing the DNA?
So, I think that the workflow varies dramatically, because when we work with modern data, we're not afraid of contaminating the sample with ourselves, with our own DNA. So, and we know that we can always go back and we can re-recruit participants. So we work with people in person. It's a different experience where we go and we collect their phenotype. So we get to interact with people. We can ask them questions. But when we work with ancient data, it's a very precious material. There's not that many ancient bones around that people are willing to give us a sample of. Yeah. And usually we have to be very careful. We have to do all the work in specific, clean conditions. So it's a very different experience, I think. I don't know which one I like more. I think I like both of them, but they're so different. And I think they provide different ways of accessing information. When we work with modern humans, we can ask them questions, but the dead cannot talk for themselves, but we can learn something from them indirectly by studying their DNA or studying their skeletal markers and archaeological evidence that contextualizes their past lives.
It was so incredibly neat. What is the process of extracting the DNA and analyzing it?
So so when we work, when we're in the field collecting samples from modern people, usually because DNA can degrade over time, we start at the extraction process in the field, and then we bring it to room temperature, and then we finish it in the lab. But when we work with ancient samples, we have to be very careful. We have to wear gloves. We have to be sure that we're wearing masks when we're taking the sample, and then when we bring it into the lab, we also have to we wear hazmat suits and masks, face shields, 2 pairs of gloves. So it's a very extreme preparation that goes into it. And then we're always so paranoid about not contaminating the sample with our own DNA, because it's intact in a way, whereas the ancient sample has degraded DNA that has accumulated damage over time. So it's not as strong as the DNA of a living person. That's why we go into such lengths to protect the sample from ourselves. And we were even joking that during the Corona time, it's we're safe because we're already using all these hazmat and like all these procedures. So
it was when I was actually thinking the exact same thing.
But yeah,
you've gotten to travel all over the world to some pretty amazing locations. Could you tell us a little bit about your travels?
Yeah. So and I kind of want to contextualize it in the study of high altitude adaptation, which is what I look at. And so to kind of give a bit of a context, there are 3 places in the world where people are exposed to high altitude and have lived at high altitude for a long time. So this is the Andean Plateau, so the Peru and I guess Chile, and then the Simian Plateau in Ethiopia, and the Tibetan Plateau. And I have done a lot of my PhD work in the Andes, and this is a picture from there. This is the Andean condor, which is a very beautiful bird. However, I didn't do my work in this beautiful location. I worked in Cerro de Pasco, which is a very high altitude location in Peru. It's at 4,300 meters above sea level, so that's not very good with feet. It's about, I think, 14,000 or 15,000 feet. And this is a mining town. What you see in the middle is this like gaping hole, which is the Cerro de Pasco mine, and people live around it. And here, as you can see, there are no trees. There's barely any plants. It's above the tree line. So it's kind of a very, it's a very, very harsh place to be for someone who's not adapted to high altitude. However, the people who live here, especially the people of the Quechua ancestry, who are the indigenous people from Peru, have genetic and physiological adaptations to this environment, which is what we were curious to study. That's why we went there and have recruited participants and collected various samples to look at that. And I was looking at their epigenetic modifications that are associated with this adaptation. And then I have also looked at the adaptation over time. And to look at that, I went to a different location in Peru, which is close to the city called Ayacucho. This is the site of Wari, which is the capital of the Wari Empire. It's a pre-Inca empire in Peru. And I was very lucky that I was able to work with these samples. They came from a mass grave, and we analyzed them for things like sex and age of death, as well as trauma. And I was able to take samples for DNA analysis. And this is the work that was funded by the Leakey Foundation that is still ongoing. So we're still working on the data and analyzing the data. We're contextualizing it, comparing this population from other populations from different time periods from Peru.
Well, when you get those results, we'll have to have you back on, and so you can share them.
I would love that. So then I did this really cool project when we went to Nepal, and this is we did the Everest base camp hike. So with a group of people from Syracuse, as well as Michigan and Sweden and Canada, we started this project by going to Kathmandu, where we collected these baseline measurements from ourselves, and then we were going to a place called Lukla. That's where all the Everest base camp hikes start. And then we did the two-week hike throughout which we were collecting samples and collecting various physiological measurements that could tell us more about how we were responding to this high altitude environment and how people were doing. So here you can see that we were collecting samples in the field. This is our first sample collection that happened in Kathmandu. And this was just in a courtyard of the hotel where we were staying there. So we were just using any space where we could. We just cleaned some tables. We tried to be clean. We tried to do the best we could with, again, a very kind of makeshift field collection type environment. And then we were staying in tea houses throughout the Everest base camp trek. So this is kind of a dining room of one of them, and you can see there's a table in the background, and it's kind of where we would also collect samples and collect phenotypic measurements. And here you can see we were collecting, here we were performing, I think, hemoglobin measurements, as well as collecting blood spots for DNA analysis. Because we were collecting saliva as well as blood to have two different sources of the DNA.
So, you mentioned that people can have some pretty severe negative effects from being in high altitude. And you were at some pretty high altitudes in your travels. Did you ever experience any of those types of symptoms?
Unfortunately, yes. I both in Peru and in Nepal, which was kind of sad. And what's even sadder, I think, for me, is that we know that once you experience high altitude associated conditions like acute mountain sickness, you are more likely to experience it again. So I know that whenever I go back, I'll probably have acute mountain sickness, and I didn't explain it, but acute mountain sickness is kind of this combination of various symptoms that people experience when they are in this environment when there's not enough oxygen. So oftentimes it's a headache, it's shortness of breath, as well as increased heartbeat. So people have a hard time sleeping through the night, because a lot of the time what happens is when you're at a very high altitude, you end up waking up in the middle of the night gasping for air. And it's a very unsettling experience. And it happens because when we sleep, we start breathing slower. So we're not getting as much oxygen. And if you combine that with being at high altitude, you're getting even less oxygen. So that then your body forces you to wake up. So what happened also to me when we were going up to the Everest Base Camp in Nepal, I got really extreme acute mountain sickness, and I actually couldn't make it up to the base camp. I think I stopped about 200 meters before we got there, but it got to the point where I couldn't make it, and I had to go down with our Sherpa guide to wait for people down at a slightly lower altitude. So, yeah, but we had an amazing graduate student with us, Taylor Harmon from Syracuse University, who helped with that, who did the last sample collection, and was really instrumental in being able to get all the samples. So that was good. But yeah, sometimes with high altitude, you don't know what might happen, and that was something that we were also interested in. Like, how can we predict it? We were collecting these samples, we were collecting these measurements to then do all these statistical analysis to see which measurements correlate with people getting sick at high altitude. Like, what should we pay attention to?
Yeah.
So yeah.
Well, thank you so much for sharing your experiences. I'll turn the virtual floor over to you now so we can take a deeper look at your research.
Thank you. So I will continue talking about my work in Nepal. So it will be about epigenetics of high altitude adaptation. And to just give more of a background, when we talk about high altitude, these are the locations in the world that are above about 2,500 meters above sea level. And if we think about various locations, so for kind of for reference, you can see about, if you look at Los Angeles, that's sea level, that's where we get all the oxygen that we need. When we were in Kathmandu, it was about 5,000 feet above sea level. The base camp altitude was about 17,000 feet above sea level. So we gained about more than 10,000 feet in about 3 weeks. And the reason why high altitude environments are difficult for people is because with higher altitude, there's lower levels of oxygen in the air. And what that leads to is various physiological adaptations that our body needs to do to be able to sustain life in this kind of environment. And the altitude, the level of altitude then affects the kind of symptoms and the kind of experiences that people have. So at high altitude, which is about 1,500 to about 3,500 meters, people experience AMS, or acute mountain sickness, as well as HACE, which is high altitude cerebral edema, and that is a potentially lethal condition. What happens also is their reaction time slows down, and other symptoms like headache can occur at this altitude. At very high altitude, which is about 4,500 meters to about 6,000 meters, learning and spatial memory can be impaired, and all these symptoms of AMS and haze can become worse. At an extreme altitude, there are issues with memory retrieval. Also, people can experience even tougher symptoms of haze and things like HAPE, which is high altitude pulmonary edema, which is fluid gathering in the lungs, which is also potentially lethal. So, kind of the symptoms that occur at high altitude are in various systems of the body. So, in the respiratory system, what happens is the breathing rate goes up. In the cardiovascular system, the submaximal heart rate goes up, as well as in the hematological system, the hematocrit goes up. So, people start producing more hemoglobin about like a week or so after being at high altitude, and overall, kind of the metabolism changes completely to adapt to this environment. And we were curious to see how this short-term exposure to high altitude can change epigenetic signatures of genes that are associated with high altitude adaptation. And to do that, we went to Nepal. So one of this Everest base camp hike was a very classic hike that we followed. So we started in Kathmandu, which is where everyone usually flies in when they want to do the Everest base camp. That's where we collected our baseline measurements. Then we collected our next measurement in Namche, which was at 3,400 meters. Then we did Ferishe, which was at 4,300 meters. And then we did Gorakshep, which is the final location, and that was the Everest base camp, and that's about 5,200 meters above sea level. And this work would have not been possible without Taylor Harmon, as I mentioned before, and she was helping with all the sample collection as well as lab work back when we extracted those samples and did the lab work in Michigan, as well as the Yaks. I guess they were also very instrumental. So carried all our equipment and they carried all our stuff. So, and they're very important at high altitude. And the reason why I was curious to look at the epigenetics was because epigenetics studies changes to the DNA that do not affect the sequence of the nucleotides. We know that various environmental exposures can affect the epigenome. So things like diet, things like exposures to chemicals, stress can affect the epigenome, as well as exercise. So we were very curious to see if this exposure to hypoxia would affect the epigenome. And the reason why was because these epigenetic modifications can change. So the modifications that we're curious in include DNA methylation. So that's here, addition of a methyl group to the DNA, as well as modifications of tails of histones. So these are proteins that DNA is wrapped around, and they can have chemical modifications that then can affect how tightly packaged the DNA is. There's also RNA interference and other epigenetic modifications. And we know that they can affect gene expression. And as I said before, environmental factors can have an effect on the epigenetic change, on the epigenetic modifications. So the epigenetic change that we were curious in was the change in methylation levels. So as I said before, it occurs at a cytosine guanine dinucleotide. So it's a cytosine nucleotide followed by a nucleotide called guanine. So here we have a cytosine that doesn't have a methyl group, and here we have a methylated cytosine, so it has this like CH3 methyl group added to it. And we know that DNA methylation here, shown as these failed lollipops, is associated with gene repression. So in this picture of a schematic of a gene, here we have the promoter region. That's where gene expression starts. And usually this promoter region for the proteins that and the enzymes that work to make expression happen, this region needs to be open. And sometimes when it's methylated, it can provide hindrance to polymerases and other enzymes adding to the DNA and being able to express it. So oftentimes methylation here can be associated with repression of various genes. So we were curious to see if hypoxia exposure affects the epigenetics, and then we wanted to connect it to also changes in the phenotype. So when looking at various altitudes, and here we have Kathmandu, Namche, Ferishe, and Gorakshep, these are the altitudes where samples were collected, we saw a change So here, every high altitude location is compared to Kathmandu, which is our baseline. So here we see an increase in hemoglobin as we went up to high altitude, which was expected. We saw a decrease in BMI, so that's what's called high altitude anorexia, where people lose weight when they're at high altitude because the metabolism changes and it switches to a system that prefers sugar. That's also why people often crave sugar at high altitude. Also, what we saw was increased blood pressure and decreased oxygen saturation. So these were all expected findings. Then we looked at various genes that are associated with how the body responds to high altitude, and these are EPAS1, EPO, PPAR alpha, and RXR alpha. And when comparing to Kathmandu over time, we saw that these genes changed their methylation levels. And I'll go through them in more detail. So here, the first one we looked at was EPAS1. It's a hypoxia-induced gene. So we know that in hypoxic conditions, it gets induced. And it's under selection in Tibetans who are adapted to high altitude. So here, I'm showing a Manhattan plot. So here, the study by Beal et al. 2010 looked at, they did a selection scan looking at the entire genome to find regions where we see signatures for selection, and EPAS1 was the one that was really strikingly different in the Tibetans, and that was the region that was associated with their adaptation to high altitude. So we were curious to look at that gene. And when we looked at the methylation levels of the gene comparing low and high altitude, we saw that the methylation levels went up the higher we went to high altitude. So then we looked at this gene called EPO, or erythropoietin. So its methylation is associated with red blood cell production. And we know that levels of EPO mRNA are significantly higher in hypoxic conditions compared to normoxia, or normal oxygen conditions. So what we saw in our sample was that there was a significantly lower amount of EPO methylation at high altitude in almost all of our participants compared to low altitude. Then we looked at genes PPAR alpha and RXR alpha, which are, they form a heterodimer. So PPAR alpha is a peroxisome proliferator activated receptor. It was identified to be under selection in Tibetans and associated with maximal oxygen uptake. What we saw was that levels of PPAR alpha were going up at the methylation levels were going up at high altitude. And then when we looked at RXR alpha, which binds to PPAR alpha and is reduced in hypoxic conditions, we saw that the methylation levels of RXR alpha were decreasing and going from low to high. And what was interesting also here, looking at PPAR alpha and RXR alpha that bind to each other, was the people who went up for RXR alpha, whereas everyone else went down, were also the people who went down for PPAR alpha. So that was another kind of another way that we saw that these two proteins could be connected. So overall, we saw that two genes, PPAR alpha and EPAS1, showed increased methylation at high altitude, while EPO and RXR alpha showed decreased methylation at high altitude when comparing Kathmandu to altitudes going up throughout the Everest base camp trek. So overall, we found, as I said before, significant changes in methylation that are associated with high altitude ascent. And I think to me, this was interesting because we're seeing now that even these short-term exposures to high altitude can affect the epigenetics, and that we were actually I think we were surprised to see that we already could see the changes in the methylation in these genes. So yeah, and with that, I would like to thank our participants and the guides who went with us. So these are the participants, and because we were participating ourselves, and our Sherpa guides who went with us, and the director of the field season, Tom Brutzart. And here I have a link to a blog that we were writing in every day. So each day, every participant would write in their blog. So, and if you're interested, you can read it. So, yeah.
And you can, we have shared the link to the blog in the chat. So please be sure to check that out. I was reading through it last night, and it was really, really interesting. Now, thank you so much, Ainash. That was an amazing talk. I'm really excited
Thank
now
you.
to get some questions from our audience. For those of you viewing live, and if you haven't gotten your questions in, be sure to get those questions into the chat now so Ainash can answer your question. I have a quick question to start us out, though. I know you've got a dream project. Could you tell us a little bit about what your dream project is?
Yeah. So I think my dream project would be to work in Kazakhstan. It is where I'm from, and I have a natural curiosity about the region. It's a country in Central Asia that is, for people who don't know, it is south of Russia and west of China. So it's kind of in the crossroads between Asia and Europe. So it's Central Asia. It's a region that has seen a lot of migrations over time, and is associated with the nomadic lifestyle and these like nomadic people who moved around a lot. So I am really curious in the history of my country, and that's and I'm working on establishing projects there. So hopefully in the future I can do some projects both looking at ancient and modern samples, ancient people and modern people back in Kazakhstan.
Awesome. Well, I think we have our first question from the audience. What was it like interacting with the people of each region?
So I think it's always different. I mean, these are different countries and different contexts. So I really like working in Peru. I think it's really just working in high altitude in Peru is an amazing thing, because when you go there, you realize how it actually is hard to live there, and people should be, and people probably are adapted. It's because when we go there and then we just feel miserable, and then we see someone just go like running down the street and be completely fine, and then just start feeling so inferior. And but I think our participants are always amazing, and they're always also curious to learn about their own biology, and people know that they are adapted to high altitude. So it's always, I think it's something that is interesting to them to see that there are these, biological, also biological not biological, but more like genetic contributions to it. And in Nepal, actually, I think it was slightly different, because there we worked on ourselves, like we were our own participants. So, but the people we were interacting with were our guides, and there too, I think you just again feel inferior, because we had Sherpa guides who did amazing, and then we were just struggling, and we were always in the back. But I mean, to them, it's a way of life. It's how, and we the guides who made it possible for us to go up to the base camp, they have their own company. This is what they do. So they are the people who make it possible for foreigners to make it to the Everest base camp and to make it to Everest, because without the Sherpa guides, it's impossible for people to make it up, because they're the ones who create the infrastructure. They're the ones who help with everything. Otherwise, I don't know if there has been any ascents without Sherpa guides being a part of it.
We have another question. This is from John Mitani. The patterns of methylation seem to show a lot of individual variation. Any ideas why? I asked because you said you didn't adapt well to high altitude.
Yeah, so we know that methylation changes are associated with that the genetics can also have an effect on the methylation. So that's why people from different populations and with different genetic signatures can have slightly different responses to high altitude. I actually excluded myself from the sample, because we just restricted it for the Nepal samples, we restricted it to people of European descent. And the same is true for when we do work in Peru. We also try to we integrate the genetic data together with the epigenetic data, so that we can control for these. I say control, but we incorporate the the effect of the genes and of the underlying genetics into our analysis, so we are not biased by this. But yeah, there are definitely changes that happen between populations, and yeah.
Do people who are adapted to living in high altitudes have problems if they move to areas of lower or even sea level altitudes?
Usually not, because the high altitude environment is the kind of the heart environment. And then oftentimes, actually, what happens, especially in Peru, and that's a big issue, is over time people can develop what is called chronic mountain sickness, and that's a condition when someone produces too much red blood cells, so their blood becomes very viscous, and they can develop cardiovascular conditions because it becomes really difficult for the heart to move the blood around. And one of the only cures is to go down to low altitude, and it goes away completely. And if people want to stay at high altitude, what they have to do is undergo phlebotomy, where some of their blood is removed. So usually people going from high altitude to low don't experience the same kind of negative consequences of that, I think. Not the same way that it happens going from low to high.
Really fascinating. But what are the differences and similarities in the genes of modern and ancient people?
So it really varies. So if we look at Peru and the context of like South America, what happened, at least between when we collected our sample, which I think they date to about 1300 AD to current time, there was the colonization of South America by Europeans. So there, the modern people in Peru have various degrees of what we call admixture. So they have genes, their genetics is mixed between the Native American component that has been passed along through generations, as well as the more recent component coming from, for instance, Spain. However, the populations that predate this pre-Columbian populations don't. So that's one of the big things where we see this shift happening through time in a place like South America. And in a place like Europe, we also see these changes happening over time. So for Europe, it's a very different context. And there, if we look all the way back to hunter-gatherers who came from Africa, and then we see this movement of European farmers, from Neolithic farmers coming from the Near East. Then we see this migrations coming from the East, from more steppe populations. So there, also these migrations have happened over multiple thousands of years and are still happening. So I think that's also for us to know, to kind of remember that people have been moving all the time. And then that's what also we're seeing in the ancient genes and ancient genetics, is that the genetics of people is not static, it's changing. And a lot of the time it's these interactions, it's these newcomers coming, populations splitting, moving. So there's a lot of change, there's a lot of stuff happening.
Yeah. We have another question. I've heard that coca leaves help cope with higher altitudes. Do indigenous peoples in these areas use it regularly?
Yes. Coca chewing is very common, and it's very easy to buy coca leaves in Peru. They're just sold in markets, on the streets. So people will just chew them at high altitude. It really helps with the symptoms. We would also drink coca leaf tea when we're up at high altitude, because it really helps with the symptoms, and it gives you this boost of energy. So yeah, it's definitely a very common thing that people do.
I think we have one more question from the audience. How did you become interested in high altitude adaptation?
Yeah, so I became interested because I'm from a city called Almaty in Kazakhstan, which is close to a lot of beautiful mountains. So I grew up looking at mountains and going hiking. So this was something that to me was it was a very it was a topic that reminded me of home, and it was also something that seeing mountains, and I grew up looking at mountains, and I think that always that aspect of it always fascinated me.
Well, Ainash, thank you so much for joining us today. And that was an amazing talk, and I really look forward to featuring you in the future when you have your research project from Peru analyzed. Again, just thank you so much for joining.
Thank you so much for having me. It was a really fun experience. Thank you.
As many of you know, this is our last episode in our pilot run of Lunch Break Science. I want to especially thank our amazing lineup of guests: Zareen, Lauren, Chalachu, Jenny, Hailai, Rachna, Dan, Anne, Caroline, and Ainash. And most importantly, I want to thank all of you, our viewers and supporters, for tuning in every week, for letting us quiz you, for trying to stump our scientists. I am thrilled to announce that Lunch Break Science has actually been renewed for another 26 episodes. New episodes will be airing live the first and third Thursday of every month, starting Thursday, October 1st. So we have an exciting new lineup of scientists and topics, and I can't wait to share details with you very soon. It's really exciting. I really can't wait. So thank you again for watching. If you want to learn more about Lunch Break Science and the Leakey Foundation, visit us at leakeyfoundation.org. You can learn how to help support research like Ainash's and programs like Lunch Break Science. Right now, all donations will be doubled by 2 generous donors. Did you miss an episode of Lunch Break Science? Visit leakeyfoundation.org/live or the Leakey Foundation's YouTube channel to catch up on episodes. Be sure to subscribe to the Leakey Foundation's YouTube channel, follow us on Facebook and Twitter, or sign up for our newsletter to be the first to hear about upcoming episodes and groundbreaking discoveries in human evolution research. Still hungry for science and can't wait till October? Check out the Leakey Foundation's award-winning podcast, Origin Stories, available wherever you listen to podcasts. Lunch Break Science is made possible by the generous support of Ann and Gordon Getty and Camilla and George Smith. Thank you all for taking a break from your day and feeding your brain with the Leakey Foundation. I look forward to seeing you all back here on October 1st. Until then, stay hungry for knowledge. Bye.